So hopefully you now have a sense that bacteria are actually really healthy. And there's a few bad apples, but the vast majority turn out to be incredibly useful. The job for this module is to learn who these superheroes are, and kind of come up with some examples that you can use to give an example of like what's a good fermenting bacteria, what's a good disease causing bacteria, what's a good disease curing bacteria. Kind of give some examples for all these valuable different types of bacteria, or at least important. Okay, so what we're going to do is go through our domains, the domain bacteria and the domain archaea. We'll do these in separate modules, and basically go through some examples. Now in the textbook it goes about, covers a lot more groups that we're going to cover, but we're going to do four major, so we have domains, below domains are kingdoms, and then within kingdoms are phyla. So really what we're talking about here are phyla. You could argue their kingdom. The concept of kind of for such an enormously diverse group becomes somewhat arbitrary, but we're going to look at kind of four major examples of bacteria, that are all thought to be generally monophyletic, um, and that basically help us understand the diversity of what it means to be bacteria. I want to remind you that in olden days, and if you take microbiology lab, there are ways to look at the metabolic properties of bacterial groups to identify them, but today, by and large, we just sequence their DNA, do a phylogeny, and figure out what kind of bacteria it is based on where they phylogenetically land. That's pretty much today we figure out the relationships, the phylogenetics, the group of bacteria that we're studying. But if you take the microbiology class in the lab, you'll learn some of the old-fashioned tricks that we still use a lot to culture bacteria and identify what they are. So the first example we're going to cover are in the phylum Firmicutes. I know it looks like firmicutes, but remember this is a formal taxonomic name, so you should pronounce it like a Greek word. It's Firmicutes. These are the Firmicutes. And here, this group of bacteria, they come in most bacterial cells are going to be either round or sometimes kind of oval shaped, what are called spheres. Or sorry, the round ones are called spheres. The elongate ovals are called rods. That's less important, but in the microbiology lab, you can see these in the microscope. Here's 10 microns. Again, you can see these are definitely less than 10 microns, classic size for a prokaryote. The key thing is the members of the Firmicutes, the big trait, and we're not going to call it a synapomorphy because these groups are so enormously diverse, but kind of pretend of it as an unofficial synapomorphy. The Firmicutes' number one trick I want you to think of them is they love lactic acid fermentation. They do anaerobic fermentation that produces lactic acid that converts a sugar to this acidic kind of sugar called lactic acid. And so they produce acid. And because of that, most of the fermenting, kind of acidifying food bacteria to make cucumbers into pickles, or cabbage into sauerkraut, or milk into yogurt. That's easy to do. You can make yogurt in like an hour if you control the temperature just right and get the right bacteria in there. Typically, they're going to make it tangy. And if you eat food that's kind of naturally tangy, but there's no vinegar in there, it's probably been produced by a firmicute. Uh, Remember the Firmicutes. So really important group of lactic acid fermenters. That's the big thing. We're going to do two examples of genera that I want you to associate with them. They're both in this clade called the Firmicutes. One, Streptococcus. This is if you want to make a judgment, this is a disease causer, so it's a bad bacteria, but really there's lots of good ones as well. This is Streptococcus, and if you've ever had strep throat, typically Streptococcus is the genus. A good way to test if you have strep throat, I'm not recommending this, I'm not a doctor. Whenever I say that, you should run and kind of ignore what I say. But if you go to the doctor and you have a really sore throat, your doctor's going to put a swab in your throat, then put that swab on a petri dish and kind of swab it and then kind of grow it either in-house or send it to a microbiology lab. And then they'll do a test. And if it turns out that it's consistent with streptococcus, then they're going to give you antibiotics, because you have bacteria that can be killed with antibiotics. If it doesn't culture as bacteria, then it's going to be something else, and they're going to say, nope, it's just a cold or a flu or a virus, and they won't give you antibiotics. So streptococcus is the cause of what's called throat. Now, if you don't want to wait to find out and you kind of want to give your doctor a heads up that, yep, it might be strep throat, kind of cough a little bit when you have this sore throat. And if your saliva, and especially your mucus, and I know this sounds gross, but again, this is the world of microbiology, if your mucus tastes a little vinegary, acidic, chances are you've got strep throat. Because again, it's producing lactic acid. And if it tastes acidic, there's a better chance that you have a bacterial infection. Okay, I'm not going to guarantee that, but again, that's usually kind of a rule of thumb. Like if I were out in the kind of woods of the world and got sick and couldn't get to a doctor, that might be a way I'd be like, okay, I need to go to a doctor. I might need antibiotics. Lactobacillus is the number one kind of producer of fermented food, whether it's kimchi, whether it's sour cream, whether it's yogurt, whether it's cucumbers, whether it's sauerkraut. Most of the fermented foods use lactobacillus. This is also the one often used in sourdough, which is really easy to make as well. You basically take some flour and water and just let it sit there and kind of come back in a few days and hopefully some bacteria gets in there. If it starts to get a little sour, you know you have lactobacillus. Okay, the name lacto is telling you not only it likes lactic acid, but also lacto is also involved in milk sugar called lactose. And so often it's converting the lactose into lactic acid is what's typically happened. So this is an important food for men. And in these slides we have it. Now, how do you identify them in the lab? Well, you could see if they become acidic, and there's lots of ways we can test for the presence of acids, like a pH strip. But the old-fashioned way is there's a stain that's widely used in microbiology called a gram stain. It should be capitalized. This isn't the measure of unit like 1 gram, kilograms, milligrams. This is named after a microbiologist named Dr. Graham. And he invented a stain in the early 20th century, it might have been late 1800s, that in the presence of bacteria interacts with the proteins in the cell membranes of certain bacteria that are called gram positives, and the cell membrane in the presence of this bacteria becomes purple. Okay, so gram positive reaction with gram stain makes it purple. So here is bacteria that's been cultured with gram stain and the cells become purple. And so again, what happens is if your doctor suspects strep throat, they culture it, they apply gram stain. If it turns purple, good chance it's streptococcus because it's a gram positive bacteria. Okay. Um, and I'll tell you how to remember purple in a second. There are some groups of bacteria that are called gram negatives, and in the presence of gram stain, they don't turn purple. They have what's called a negative reaction. They have a gram negative reaction, and here they turn pink. And there are some bacteria that don't react at all, that don't turn purple or pink, and they don't really have a name. They're just gram, maybe neutral. They don't have a reaction. Okay, here's the way to remember, is that pink, the third letter is an N, it's negative. The third letter of, or sorry, the fourth letter of purple is P, that's positive. Okay, so that's one way to do it. That's what our microbiologist at campus recommends is purple positive, pink negative. The way she says it is purple positive, pink negative. Okay, the way I do it is a little bit different. But whatever works for you, remember that. You don't want to go by the first letter, purple and pink, because purple and pink both start with a P. That's not going to help you. Okay, so remember a positive reaction is purple, a negative reaction is pink. Okay, although it looks reddish, but we call it pink. This group of bacteria, the gram negatives, includes a group called the proteobacteria. And this group here doesn't have a standard metabolism. This is an incredibly metabolically diverse group of life, also enormously diverse, so it's hard to characterize it. But the important thing is there's a really important species named Escherichia coli, and yes, that's a bugger of a genus name, Escherichia or Escherichia. Pronounce it either way. This is a bugger to spell, so people pronounce it instead abbreviated E. coli. And E. coli is sometimes bad. It's the number one cause of food poisoning on Earth. I'll tell you how you get that in a second, but you can pretty much guess. It's also the world's most important model organism for understanding how prokaryotic cells live and how they do things. If you're working in a microbiology lab, you are growing E. coli. It's also one of the most common bacteria in your gut. It's super common. It's in your gut, sometimes on your skin, especially after you've gone to the bathroom, because it's going to be in your environment. The way food poisoning typically happens, there's lots of ways to get food poisoning, but the most common, if you ever hear an outbreak at like a food or a restaurant, or in lettuce, is typically, if it came from a restaurant, somebody working with the food, with raw food at the restaurant, goes to the bathroom, they don't wash their hands. It doesn't mean they had feces on their hand. Okay, it's not like they were being disgusting. Okay, like if they don't wash their hands and they have feces on their hand, and then they cook, yeah, that's terrible. You're going to get food poison. But what it mostly is they walk in the bathroom. They could just walk in for a few minutes and then walk out. Their body's now covered in E. coli, including their hands, and they didn't wash their hands. It's not that they intended to get feces or just have terrible hygiene. It's that they didn't wash their hands. It's on their hands now, and then they cook. When you work in a restaurant or with food, whether it's at home or at a restaurant, every time you leave the bathroom, no matter what, wash your hands and then work with your food, and that keeps the E. coli off your hands. It's still on your body, but again, it's not on your hands, which is what the food's in contact with. That's the most of it. If you're getting it from like lettuce and there's an outbreak in the food at the grocery store, typically what happens there is at the factory where they are cleaning it and bagging it, it got contaminated there. Or if it comes from the farm, a farmer had some cow manure nearby. They had cows or horses or pigs or something nearby, and it rains, and some of the manure washes into the water that is used to spray to grow the vegetables, and then they don't wash it adequately, and so some of that manure E. coli gets on the food, and then if you don't cook it or wash it properly, then you ingest it. That's typically E. coli that's responsible for that. Okay, so good thing for biochemistry, for biotechnology especially, but also potentially a disease causer. Okay, we've got gram positive, gram negative. Let's do two more. Actinobacteria. This is the world's most common bacteria in soil. So that's what I want you to think of them. And so they're incredibly important in nitrogen fixation and all these chemical processes and decomposition and recycling that occur in soil, important for climate change. They look a lot like the other ones. And if you apply gram stain, this should be capital G, they are gram positives, which if it's, you remember, gram positive turns what color? You got it. Gram positive is purple. Okay, the color here is not a good color for this. So in the presence of gram stain, they turn purple. Um, the important example I want you to know is the genus Streptomyces. Streptomyces is the world's most important, really common bacteria for most of the antibiotics that we use. If it ends in MYC in the streptomycin, vancomycin, if the word MYC is in the antibiotic, chances are some biologist somewhere figured out how to steal that chemical from Streptomyces. There are other bacteria that produce antibiotics, but this is by far the world's most important. So think of them as nitrogen-fixing soil inhabitants that also often produce antibiotics. Actinobacteria, really important. I should point out that the bacteria that are gram-positive and gram-negative, that's not a clade thing, that's not a synapomorphy. It's kind of somewhat convergent. They just have similar kinds of proteins that react with the stain. Okay, and the last group, oh, I forgot to mention this. I should quick mention the proteobacteria often have weird things that look like cilia and flagella. They have different names for bacterial cells, but often these cells can move around, which is kind of weird. They can actually kind of crawl, which is a weird thing to think of individual cells crawling around. Okay, the final group of bacteria, we're not going to give it a genus. There's a bunch of different genera that exist, but this is a group of bacteria called the cyanobacteria. And this group of bacteria have the interesting characteristic that many of them, not all, but many of them live in water. Most of the rest lived either on skins or soils. This one tends to live in water, super common. If you get a glass of water, let it sit out, chances are some cyanobacteria will get in there. And if you look, the cells are green. And that tells you that they do a different type of metabolism. They're going to do photosynthesis. So a lot of these do photosynthesis. Some do nitrogen fixing as well. And evolutionarily, we think this is one of the earliest groups of life to exist, and they're responsible for the first photosynthesis on Earth, the first oxygen in the atmosphere. They're very important, and they're more common. They can be in soil, especially if it's moist, but they're way more common in water. Really, really common. And the name cyano basically refers to the color blue-green that's photosynthetic.